Hi everyone. Today we talk about chronic kidney disease or CKD. And chronic kidney disease is not a single disease. It's a progressive, self-perpetuating process in which structural and functional damage to the kidney accumulate over time. And regardless of the original trigger, if it's age, genetics, infection, immune-mediated injury, or toxic exposure, CKD follows a remarkably consistent biological trajectory. And the process usually begins with the damage in a subset of nephrons, the functional unit of the kidney. And as the nephrons are lost, the remaining ones try to compensate by increasing the filtration pressure. And this adaptive hyperfiltration initially, preserve the kidney function. So this is fine, but it comes at a cost. And in fact, over time, this elevated intraglomerular pressure accelerates the glomerular sclerosis, and this leads to further nephron loss. And as CKD progresses, then injury extends also beyond the glomerulus. The tubular epithelial cells experience chronic stress from protein overload, from the oxidative damage, and from the impaired oxygen delivery. And this triggers inflammation signaling, fibroblast activation, progressive tubulointerstitial fibrosis, and basically you get to a point where the fibrosis is really just blocking the kidney and the filtration process is impaired. And at this stage, kidney damage becomes largely irreversible. But CKD is not confined to the kidney. Persistent low-grade inflammation alters the cytokine signaling, the immune cell behavior, and then ultimately also the endothelium function. The metabolic waste that gets produced then accumulates, and it leads to uremic toxicity, oxidative stress, vascular dysfunction, electrolyte imbalance, altered calcium phosphate metabolism, and hormonal dysregulation, which then in the future, further increase the tissue damage. And this complex, multi-layered pathogenesis is exactly why CKD cannot be modeled by just one single biomarker or acute injury assay. And the samples from dogs, which naturally occur in CKD, they can be used to capture this progressive neuron loss, this chronic inflammation and fibrosis, the altered drug clearance and exposure, if this is obviously used in these kind of studies, and then basically the evolution of the disease in its different phases. And for this reason, the CKD samples from dogs with CKD are not just used in veterinary research, but they are also used in human preclinical studies and in human preclinical drug development. For example, they enable to set up assays for assessing organ damage and nephrotoxicity before a compound is then moved into the vivo studies, for example, with beagle dogs. And this obviously improves risk assessment and translational relevance. And if you want, you avoid possible mistakes and repeating animal studies, which obviously slow down the development quite significantly and cost a lot of money. So if you want to explore on your own a little bit more, what are the common assays that are normally performed for the different types of samples, being these plasma, serum, urine, PBMCs, or other kind of cells or whatever, then I've left a page here in the links, probably will be in the description or in the comments, which some more information. So you can get lighter this information than spending times and hours on PubMed. Having said that, thank you very much for watching. I hope this was useful. Bye.